Hello everyone. Today we're going to talk about the concept of communication between cells and chemicals. We're going to talk about how those chemicals bind to specific receptors within the cell or on the cell's surface. So any signal molecule, or we can refer to it as a ligand, those signal molecules could be anything from hormones, proteins, odors, even drugs that we take. Those signal molecules, they usually bind to specific target receptors, and those target receptors are either found on the surface of the cell, or those receptors would be located inside of the cell, either within the cytosol, that fluid inside of the cell, or actually inside the nucleus. Now, where a chemical binds, whether the chemical binds to the receptor on the surface or whether it binds to the receptor inside of the cell, that actually depends on whether that chemical or that signal is lipophilic or lipophobic. So lipophilic, meaning that those molecules, they love lipids. They are able to dissolve within lipids. Lipophilic molecules, right here, those lipophilic signal molecules, they usually diffuse very easily through the plasma membrane. So if this is a cell right here and this is the plasma membrane, if you remember the plasma membrane, it's a bilayer, and this middle layer right here of the plasma membrane contains lipids, fats. So it makes it easier for a molecule that's lipophilic to pass easily through the fatty layer of the cell membrane. And so an example of some lipophilic ligands would be, we have some hormones that can pass easily through your plasma membrane. And then once they are inside the cell, they will bind to the receptors, and those receptors, they're usually either in the cytosol or they can bind to receptors that are found deep inside of the nucleus. And once they bind, let's say, to the receptors found inside of the nucleus, they can actually trigger the activation of certain genes, so they can turn them on. And those genes, after they get turned on, the process of transcription and translation can go ahead and take place, and they can and proteins would start being made. And usually this process could take about an hour or even longer for you to get the final results. Or they can actually inactivate certain genes and stop the production of certain proteins. Now, on the other hand, if you have a ligand and that ligand is lipophobic, lipophobic meaning that they do not like fat. So those would actually have a hard time passing through the plasma membrane directly because of that lipid layer right here within the plasma membrane. So usually lipophobic molecules, they go ahead and they bind to the receptors located on the surface of the cell. Once they bind to the surface of the cell, certain, mechanism will take place, several steps will take place. We'll talk about actually an example of those ligands that bind to the surface receptors, and we'll talk about the pathway that takes place. And the response, you can actually see it within milliseconds to minutes. It's a bit faster, actually, it's way faster compared to the activation or inactivation of the gene response. This image right here is showing you an example of different membrane receptors. So as we said, if you have a signal molecule, That signal molecule could either be a hormone, a protein, any, like, there's like a variety of signal molecules. Those signal molecules to go ahead and cause a response within your cell, they would have to bind to receptors. Some of these receptors are channels, receptor channels, where they have a specific site for that ligand to go ahead and bind to it. Once it binds to it, it usually activates that channel, opening that channel or closing it, either allowing things to pass through or closing it, preventing certain things from passing through. Some of these receptors that the ligands would bind to it, notice how this receptor has a very unique shape. It usually kind of twists like that within the plasma membrane, and it twists actually 7 times if you count, like 1, 2, 3, 4, 5, 6, 7. So they usually call it the 7 membrane receptor. Usually these receptors right here are bound or coupled to a protein called the G protein. And once a ligand binds to this receptor, that G protein gets activated, causing the activation of other proteins inside of our cell. We are going to focus on some examples of those G protein coupled receptors right now. This is just showing you how some receptors are also linked. Some of them are also linked to enzymes. So once that receptor is activated by the binding of the ligand to it, that enzyme gets activated and can cause, again, activation of some internal enzymes, and the response would, different responses could take place inside of the cell. So let's go ahead and concentrate on those G protein coupled receptors and take a look at an example. So here is the receptor, right? The one that twists, as I said, 7 times within your plasma membrane. And we call it a GPCR. Why? G is for the G protein. So G protein coupled receptor. So it's a receptor that's usually coupled, that's basically bound to a protein known as the G protein. Now, we'll go ahead and take a look at what happens when this G-coupled receptor is activated. This right here is a signal transduction image. And first of all, you might be wondering what signal transduction means. It just means a process that's going to take place when a ligand activates a receptor, which in turn is going to cause alteration in your intracellular molecules, creating a response. So it's a kind of like a domino effect, okay? Several enzymes and several like proteins get activated, causing basically a response at the end. So this G protein coupled receptor, we refer to it as G protein coupled receptor adenylyl cyclase, and some ligands that go ahead and bind to this specific G protein coupled receptor is epinephrine, or basically adrenaline. So for example, we have that G protein coupled receptor on the surface of the heart cell. And so when epinephrine binds to it, it causes your heart contraction to increase. So it speeds up your heart contraction. We also have some of these receptors within our intestines. And so again, we'll talk about how epinephrine would bind there and affect the motility of your GI tract. So let's see exactly what happens when those ligand molecules bind to that specific receptor. So once a ligand binds to that receptor, as we said, an example is epinephrine, that G protein will go ahead and get activated. So the G protein will only get activated if the ligand binds to the receptor. Okay, so if the ligand does not bind to the receptor, that G protein stays inactivated. So once a ligand binds to that receptor, and it has to be a specific ligand, right? Because it will have to fit to that receptor. So not just any ligand can go ahead and bind to any receptor. There has to be a match, okay? They have to be a match. So once they bind to that receptor, the G protein gets activated. Once the G protein gets activated, it will go ahead and turn on an enzyme. This enzyme is known as adenyl cyclase. Once adenyl cyclase gets turned on, basically activated, it will go ahead and convert some of the ATP that you have inside of your cell to a compound known as cyclic AMP or CAMP. CAMP, we refer to it as the second messenger. So our first messenger was the ligand that started this cascade event to take place. So this is the first messenger right here, and we refer to the cyclic AMP as our second messenger. The cyclic AMP or cyclic AMP, it's basically a compound made up of adenosine, or like the word stands for cyclic adenosine monophosphate. So it has a phosphate, it has adenosine. Basically, it has a bunch of carbons, hydrogens, nitrogens, and an oxygen and phosphate. So this cyclic AMP is a second messenger that's going to activate a bunch of protein kinases. And we refer to them as protein kinase A because we have several types of kinases. Whenever you see the term kinase, you know that this protein is going to help phosphorylate, which means it's going to add a phosphate. So we have a bunch of different ones. This one right here is known as protein kinase A. So protein kinase A gets activated when cyclic AMP gets activated. Adenyl cyclase, sometimes they refer to it as an amplifier enzyme, by the way, because it's going to activate several molecules. And those several molecules, once they're activated, they're going to activate some other molecules, so the response is amplified. That's why they call it an amplifier enzyme. So just to recap one more time what we've discussed, you have a ligand molecule. It will bind to that G protein coupled receptor. Once it does bind, the G protein will get activated. Once the G protein gets activated, it's going to turn on an enzyme, activating that enzyme, which is our amplifier enzyme known as adenyl cyclase. Adenyl cyclase will go ahead and take some of the ATP inside of your cell and convert it to cyclic AMP, which is going to act as our second messenger. Once cyclic AMP gets activated, it will go ahead and activate a bunch of enzymes within your cell known as protein kinase A. What are those proteins going to do? Well, they're going to go ahead and start phosphorylating a bunch of proteins you have inside of your cell. So they're going to start adding phosphates to proteins inside of your cell. Once those proteins are phosphorylated, the cell will go ahead and have a response. So as I said, one of the responses that would take place would be when epinephrine binds, and this whole pathway takes place when epinephrine binds to the surface receptors on your heart. The response would be that your heart rate would increase. Okay, now depending on where you have that G-coupled receptor, whether it's on the surface of your heart, whether it's on the cells of your intestines, of course the cell response is going to differ.